Hello dear students, welcome to the biology online class. So this video is the continuation of chapter 2, that is sexual reproduction in flowering plants. So in the previous class we have studied about the male reproductive organ of the flower, right? So today let us take up a new topic, that is the female reproductive organ of the flower, which is also called as gynoecium. So as I said before, this gynoecium, it is the female reproductive part and it is also called as pistil or carpel. And based on the number of carpels, the pistil may be classified into two types. They are monocarpellary pistil and multicarpellary pistil. So once again I will repeat, based on the numbers of carpels, the carpels are divided into two types. They are monocarpellary pistil and multicarpellary pistil. So if in a flower it contains only one pistil, then it is called as monocarpellary. So monocarpellary means presence of one pistil, whereas multicarpellary means if the flower contains more than one pistil, then it is called as multicarpellary. So in the term multi means many. Am I clear? So this is the classification of pistil based on the number of carpels. Am I clear? Okay. Now in multicarpellary, the pistils may be fused together or free. Okay. If the pistils are fused, then the condition is called as syncarpus. Whereas if the pistils are free, then it is called as apocarpus. Okay, if the pistils are fused together, then the condition is called as syncarpus. Here syn means fusion. Whereas if the pistils are free, then the condition is called as apocarpus. Am I clear? Okay. Now coming to the parts of a pistil. Each pistil, it has three parts. They are stigma, style and ovary. Now stigma, it is the tip portion of the pistil and it is the landing platform for the pollen grains. So in the picture, this part is called as the stigma. Okay, so this stigma, it is the tip portion and it also receives the pollen grains. Okay, now next to the stigma, the elongated portion is called as the style. So style is elongated slender part beneath the stigma, and the basal swollen portion is called as the ovary. Okay, so in this way each pistil consists of three parts: stigma, style and the ovary. Stigma is the tip part, style is the middle part and ovary is the basal part. Okay, so now within the ovary it contains a chamber, and that chamber is called as ovarian cavity or locule. Am I clear? So within the ovary there is a locule. So now this portion is the ovary, which is a bulged portion, and within the ovary the chamber is called as the locule or ovarian cavity, and in the locule placenta is present, and from the placenta many ovules are produced. Am I clear? So inside the ovary is the ovarian cavity, which is also called as locule, in which placenta is located. Arising from the placenta are the ovules, which are also referred as megasporangia, and the number of ovules in an ovary may be 1 to many. So in this picture you can clearly observe the locules. What do you mean by locule? Locule is nothing but It is the ovarian cavity or the cavity present within the ovary. Now in the locule, placenta is present. This portion is the placenta, and from the placenta many ovoid bodies are produced, and these ovoid bodies are nothing but the ovules. Understood? So and each ovule contains one to many each ovary contains one to many ovules. Am I clear? So this is the enlarged portion of the ovary in which we can observe the locules, the placenta and the ovules. Any doubts? Clear? So these are about the parts of a pistil. Now coming to the structure of megasporangium. What do you mean by megasporangium? Megasporangium is nothing but ovule. So ovule which is present within the ovary, it is also called as megasporangium, right? Now coming to the structure, ovule is attached to the placenta by a stalk called funicle. So here funicle is called as the stalk of the ovule, and the junction between the body of the ovule and the funicle is called as hilum. Okay, in this picture you can clearly observe the funicle as well as the hilum portions. So this is the structure of an ovule. So in this you can observe the funicle region as well as the hilum regions. So funicle is nothing but, this is the stalk of the ovule. So this stalk like portion is called as the funicle, and with the help of this stalk the ovule is attached to the placenta. Am I clear? And now the ovule, it mainly contains two portions, the stalk portion as well as the main body. So the stalk portion is here called as funicle, right? Now the junction between the stalk and the main body, so this entire portion is nothing but the main body of the ovule. Am I clear? So the junction between the funicle and the main body of the ovule is called as hilum. So this portion is the hilum. Understood? So hilum is nothing but, it is the junction or it is the place between the funicle as well as the main body of the ovule. Am I clear? Okay, so this is about the hilum and the funicle. Now each ovule, it has one or two protective envelopes which are referred as integuments, except at the tip where a small opening called micropyle is present. And opposite to the micropylar end is the chalaza, which is nothing but the basal part. And enclosed within the integuments there is a mass of cells called nucellus, and the cells of the nucellus contain reserve food materials, and inside the nucellus is the embryo sac, which is also called as female gametophyte. And in the embryo sac three groups of cells are present. They are antipodals, egg apparatus and secondary nucleus. Okay, now coming to the structure once again. Here the stalk of the ovule is called as funicle. Okay, so what is the meaning of hilum? So hilum is the junction between the funicle and the main body of the ovule. Correct? Now each ovule, it contains one or two protective layers, and these layers are referred as integuments. So in this ovule, it contains two integuments, the outer integument, this one is the outer integument, and this one is the inner integument. Understood? So the ovule consists of a protective layer which is called as integuments. Now this integument, it do not cover the ovule completely, but it leaves a small area uncovered, and that uncovered area, it is in the form of opening, which is called as micropyle. So micropyle is nothing but it is the small opening of the ovule which is present at the tip. So micropyle is the tip region of the ovule. Now just opposite to the micropyle there is a basal portion which is called as chalaza. So opposite to the micropyle, chalaza is present, and this chalaza, it is the basal part of the ovule. Am I clear? Now, next to the integuments, the ovule contains a mass of nutritive cells which is called as nucellus. So here nucellus means here the cells of the nucellus contains, the nucellus tissue contains mass of cells, and the function of the nucellus is to provide nourishment to the developing spores. Understood? Now next to the nucellus there is an embryo sac. So here the embryo sac is also called as female gametophyte. Once again I'll repeat. Next to the nucellus there is an embryo sac, and this embryo sac is also called as female gametophyte. Why it is called as female gametophyte? Because this embryo sac carries the female gamete. So hence it is called as female gametophyte. Am I clear? Now, in the embryo sac, 3 groups of cells are present. Antipodal cells which are present at the chalazal end, then egg apparatus cell which is present at the micropylar end, where it consists of single egg cell and 2 synergids, and at the center it contains a secondary nucleus. So in this way 3 groups of cells are present in the embryo sac. So these are about the structure of megasporangium. So once again I'll repeat the structure. Each ovule, it contains the stalk portion which is called as funicle, and the junction between the funicle and the main body of the ovule is called as hilum. The protective layer which is present in the ovule is called as integuments, and the integuments is divided into 2 types, outer integument and inner integument, and the tip portion of the ovule is called as micropyle which is in the form of opening, and just to the micropyle, just opposite to the micropyle, there is a basal portion which is called as chalaza, and next to the integuments there is a nutritive tissue which is called as nucellus, and in the nucellus there is an embryo sac which is also called as female gametophyte, and in the female gametophyte it contains 3 groups of cells. At the chalazal end antipodals, and at the micropylar end it contains egg apparatus. In the egg apparatus it contains 1 egg cell and 2 synergids, and at the center it contains secondary nucleus. Am I clear? So these are about the structure of megasporangium. Okay. Now we'll move on to the next concept, that is megasporogenesis. The process of formation of megaspores from megaspore mother cell through meiosis is called as megasporogenesis. So once again I'll repeat the definition. Megasporogenesis means it is the process of formation of megaspores from the megaspore mother cell through meiosis. Okay. So usually what happens is a single megaspore mother cell in the micropylar region, the ovule generally differentiates a single megaspore mother cell in the micropyle region of the nucellus. And this megaspore mother cell, it has a dense cytoplasm and a prominent nucleus. And later on, this megaspore mother cell undergoes meiosis to produce the megaspores, and 4 megaspores are formed during the megasporogenesis. Okay, so coming to the explanation. So during the developmental stages, what happens is any cell of the nucellus, that means in young ovule there is no embryo sac. That means next to the nucellus, usually the mature megasporangium contains the embryo sac or the female gametophyte. Within that it contains antipodals, secondary nucleus and egg apparatus and all. Correct? But in young ovule, the embryo sac is entirely absent. That means the female gametophyte is entirely absent. Okay. So later on what happens? Any cell of the nucellus at the micropylar region. Once again I'll repeat. Any cell of the nucellus at the micropylar region, it becomes enlarged and it gets converted into megaspore mother cell. Am I clear? So during the developmental stages, any cell of the nucellus at the micropylar region, it becomes enlarged in size and it gets converted into megaspore mother cell. Okay. Now, all the cells of the nucellus are diploid in nature. Okay. So, what happens here? This megaspore mother cell is also diploid in nature. Why? Because it develops from the nucellus tissue. Am I clear? So, now this diploid megaspore mother cell, it undergoes meiosis. Okay. So the megaspore mother cell which develops from the nucellus, it undergoes meiosis. As a result of this, 4 megaspores are formed, and all these megaspores are haploid in nature. Correct? Okay. So now this is the portion of megaspore mother cell which develops from the nucellus tissue, and this megaspore mother cell, it is always diploid in nature. Okay. Now later on, this megaspore mother cell, it undergoes meiosis. As a result of this, 4 megaspores are formed. In this way, 4 megaspores are formed, and all these megaspores are haploid in nature. Correct? And this process is called as megasporogenesis, means the formation of megaspores from megaspore mother cell through meiosis. Am I clear? Any doubts? Clear? Okay. So in this way, from a single megaspore mother cell, 4 megaspores, haploid megaspores are produced. Okay. Now coming to the next topic, formation of female gametophyte. How this embryo sac is formed. Okay. Now in majority of the flowering plants, 1 megaspore is functional, while the other 3 degenerate. And the functional megaspore develops into the female gametophyte. The embryo sac formation from a single megaspore is called as monosporic development. Okay, so this is the picture of megasporogenesis. So as well as the formation of female gametophyte. So here what happens during the first case? So this is the megaspore mother cell which is formed from the nucellus tissue. Later on this megaspore undergoes meiosis. As a result of this megaspores, tetrad is formed. 4 megaspores are formed. Till here the process is called as megasporogenesis. Now after megasporogenesis, so of these 4 megaspores, the 3 megaspores degenerate while only 1 becomes functional. Now this is the functional megaspore with its haploid nucleus. Am I clear? Now this functional megaspore, it becomes the female gametophyte or the embryo sac. Understood? Okay. Now, the nucleus of the functional megaspore divides mitotically to form two nuclei. They move to opposite poles, forming two nucleate embryo sac. The nuclei again divide two times, forming four nucleate and eight nucleate stages of the embryo sac. And these divisions are free nuclear. That means nuclear divisions are not followed immediately by the cell wall formation. After the 8 nucleate stage, cell walls are laid down, leading to the organization of the typical female gametophyte. And 6 of the 8 nuclei are surrounded by cell walls and organized into cells. And the remaining 2 nuclei, which are also called as polar nuclei, are situated at the center, form the secondary nucleus. And a typical mature embryo sac is 8 nucleate and 7 celled. This is important. And 3 cells are grouped at the micropylar end and form the egg apparatus. It consists of 2 synergids and 1 egg cell. And the synergids have special cellular thickenings at the micropylar tip, which is called as filiform apparatus, and it helps to guide the pollen tubes into the synergids. Okay. The 3 cells at the chalazal end are called the antipodals, and the large central cell called the secondary nucleus, which has 2 polar nuclei. So once again I'll repeat the process. Now after megasporogenesis, from a single megaspore mother cell, 4 megaspores are formed, right? And all these 4 megaspores are haploid in nature. Okay, later what happens? Out of 4 megaspores, the 3 megaspores degenerate and only 1 megaspore will becomes functional, correct? And this functional megaspore, it becomes the embryo sac or the female gametophyte. And now this functional megaspore, it contains single nucleus which is haploid, correct? Now later on, the nucleus of the functional megaspore, it undergoes mitotic division. Once again I'll repeat, the nucleus of the functional megaspore, I mean the nucleus of the female gametophyte, it undergoes mitosis division. As a result of this, two nuclei are formed in this way. Okay, so two nuclei are formed. Later on, one more division takes place, so that 4 nuclei are formed, and later on third division takes place. So in this way 8 nuclei are formed. So now within the embryo sac, it contains 8 nuclei. Now of these 8 nuclei, the 3 nuclei move towards the chalazal end, and by the wall formation it gets converted into antipodals. Once again I'll repeat, of 8 nuclei, the 3 nuclei move towards the chalazal end, and by the wall formation it forms the antipodals. Okay. Now another 3 nuclei move towards the micropylar end, and it forms the egg apparatus, where it consists of single egg cell and 2 synergids. Am I clear? And the remaining 2 nuclei are located in the center, which forms the secondary nucleus. And this secondary nucleus, it has 2 polar nuclei and is always diploid in nature. Understood? Is that clear? So in this way, at maturity, the embryo sac contains 7 cells and 8 nuclei. Okay, so it is called as 7-celled 8 nucleate embryo sac. Okay, and in the synergids, it contains special cellular thickenings which is called as filiform apparatus. And what is the function of filiform apparatus? It helps to guide the pollen tube into the embryo sac. Understood? So these are about the formation of female gametophyte. And this is the picture of a mature embryo sac, where you can see the three cells which at the chalazal end forms the antipodals, and at the center it forms the secondary nuclei. That means the secondary nucleus, it consists of two polar nuclei, and at the micropylar end it consists of egg apparatus, where it contains a single egg cell. So this is the egg cell, and two synergids, and in the synergids it contains filiform apparatus. Okay, so this is about the explanation of 7-celled 8-nucleate embryo sac, or the development of female gametophyte. So these are about today's topic. Let's continue in the next class. Thank you.